VIPER, take a privilege of introducing the speaker of this session, Dr. Vishwanath Buniyad, sir. Dr. Vishwanath Buniyad received PhD in 2012 from National Institute of Technology, Durgapur. He completed his B.Pharm and M.Tech in Biotechnology from Jadavpur University, Kolkata. He received DST Fast Track Young Scientist Award and CSIR Research Associate Fellowship in 2012. Following completion of his PhD, Dr. Buniyad joined to academia as Assistant Professor in the Department of Bioengineering at National Institute of Technology, Agartala. His research area is focused on bioprocess engineering, environmental biotechnology, pharmaceutical biotechnology, and bioinformatics. He is providing his services as visiting faculty to various colleges. He is acting as editorial board member, guest editor to various journals indexed in Bentham and Scopus. Dr. Buniyad is also acting as reviewer for various Elsevier journals. He has authored and co-authored 70 peer-reviewed papers, 15 book chapters, 1 book, and presented at numerous national and international conferences on topics ranging from bioprocess engineering, bioinformatics, enzyme and microbial technology, advanced purification process, and modeling. With this brief introduction, I like to invite Dr. Vishwanath Buniyad sir to take the session.
Okay, good afternoon to all of you. Thanks, Dr. Satya, for your warm introductions. I'd like to thank organizer, faculty member of the department, Vishnu Institute of Pharmaceutical Education and Research, Telangana, especially Dr. K. Bonita, Professor and Head, Department of Pharmaceutics, VIPER, and Dr. A. Ramesh, Principal, VIPER, and Dr. Ved Thierry, Principal Scientist, ICRI, New Delhi, for giving me an opportunity for sharing my thought in 5 days faculty development program, recent advances in science and technology innovation through online mode. I welcome all the participants, including faculty members, scientists, students, and researchers for this session. Let me allow to ask, sir, am I audible?
Yes, sir.
Madam, allow to present the PPT because it is showing that host disabled participant.
Yes, sir. Actually, you have rejoined. That is the reason. Rahul, 1 minute, sir. Now you are the co-host, sir. You can share.
Okay, that's fine. So today I thought I will share my views on the recent development in production, purification, and characterizations of the microbial enzymes, since it is matched with your theme that recent advances in science and technology innovation. First of all, we need to know that what is enzymes. Enzymes, I know everybody that it mostly catalyze the most of the biochemical reactions. But enzyme basically that is the chain of the amino acids, and that amino acids are linked with the peptidyl bonds, and the peptidyl bonds create the primary structures. But three-dimensional structures of the enzymes denotes the function of the enzymes. If you enzymes maintains its integrity of the structure, then only it can catalyze a specific type of reactions. Now, if you think about the enzymes, it is different from other chemical catalysts or not. So definitely you have to understand the mechanism behind the catalytic reactions to be conducted by the enzymes. Enzymes is very specific particular reactions, and that can be catalyzed by them. Enzymes nomenclatures, or its name is given what catalytic reactions it can be conducted. If it is done it is a protease, then definitely it break down the proteins. That's why its name is protein plus ase, protease. And obviously, if it is degrade the lipids, its name will be lipase. Beauty in the enzymatic reaction is that it is a speed of the rate of reactions, that any conversions up to 10 to the power 24, which may not be possible by the chemical reactions. If you are talking about a reaction which is going to happen in the chemical way, where sulfur dioxide plus oxygen is going to react and form the sulfur trioxide, where vanadium pentoxide was used as a catalyst. If you compare the biocatalyst with chemical catalyst, you will see the speed up of the reactions can be happens up to 10 to the power 4 level for the chemical reactions, but biochemical reaction is very, very fast, but specific for specific reactions. As usual way that it follow the characteristics of the catalyst, that means it never participate in the reaction and never consumed, and never change the equilibrium of the reactions, never change the thermodynamics also. But it only reduces the activation energy of a particular reactions. If you compare with the chemical reactions, the activation energy reduction is quite lower than what enzyme can do. It happens due to the hydrogen bonds and several van der Waals forces acts upon the substrate or reactants. Now, if you think about how the enzyme works, and it's happens in the aqueous phases, where enzyme gets solubilized. If the enzyme is solubilized in the butters, that having some hydrogen bonds which makes the enzymes stable and soluble in butters. The hydrophilic amino acid, which is present on the surface of the three-dimensional structure, that creates a hydrogen bond with the butters molecules, and that helps to make it stabilize and stabilize in the butters. Now, if somehow you change the environment of the proteins molecules, then it loses its integrity in the structure and it becomes dysfunctional. Have a look how enzymes basically works in particular reactions. If you see that in the x-axis, there is a progress of reaction as a path of reactions. In the y-axis, there is an energy. A reactions will happen or not in presence or absence of the catalyst, that only be, you can say, on the basis of Gibbs free energy. And the Gibbs free energy is that the energy exists in the reactants and difference between the reactants and products energy. This energy is called the Gibbs free energy, and that if the reaction to be proceed, you need to give some energy to molecule to be transferred to the transition states, and that energy is called the activation energy. And what enzyme do basically, enzyme never alter the Gibbs free energy, it only reduces the activation energy and it's a larger extent. Now, classification of enzyme, there are lots of debate, but the enzyme commissions, what they have classified the enzyme in the 6 broad category, and the most important section is the hydrolase. That enzyme needs water molecule for specific catalytic reaction and is exploited a lot in industrial scale. It around 80% of the total enzymes markets captured by this hydrolase, and it needs water to break down the reactions. These are hydrolytic enzymes. These are the amylase, protease, pectinase, xylanase, the all enzymes under this category. Although there are several other categories enzymes are there: oxidoreductase, transferase, lyase, isomerase, ligase. Ligase is very important enzymes in the molecular biology. It's covalently joined the two molecules and the energy came from ATP. So these are the classifications of the enzymes. Now, one more important part when enzyme is produced in the systems or enzyme is extracted, you can do from any cells. Then ultimate question is, what is the activity or how much enzymes you have extracted? What is the quantitative informations? So that you can only understood by doing its catalytic measurements. The unit of enzymes is expressed in the form of product formation rate. If an enzyme acts as a catalyst for a particular reaction where reactant is converted to product, then dP by dt is the product formation rate in reaction engineering perspective. And it is in biochemistry is called the V, that is the velocity of the reaction. And it is nothing but the product of rate constant and enzyme concentration. You know that rate constant, that is sometimes called the turnover number of the enzymes, and it depends only the temperature and pH or environmental parameters can change the turnover numbers. It never depends the concentrations of enzymes, what is used, and that is the beauty of the rate constants. And you know, the most of the biochemical reactions are first order reactions, and that's why that power of the enzymes concentration is 1. If we go for any design of that any catalytic reactions, you have to know what is the rate constants value, and that is the measurement of unit of enzymes to be expressed or to be extracted or produced by a microbial systems. Now, if you see the market, global markets of the enzymes, it is around $8,636.8 million. That is in 2019's report, and is expected to be enhanced $14,507.6 million in 2027. Now you imagine that how much market contributed by that industrial enzymes. Yet we are in lag stage. Most of the enzymes we exported from the other countries. Although few industries are making these enzymes, if you see the global scenario, carbohydrates segment are the dominating sector in 2019, to be expected will be in 2027 also. And if you see that microorganism is a sector where the enzymes are mostly used as a source for enzyme extraction or a enzymatic process development. Now, if you compare with the plants and animals, they are in very less amount contributes. Figure 3 illustrates the importance of the hydrolase, what I have told initially. 70 to 80% of the enzyme market contributed by the hydrolase enzymes. And that is given in shown in the figure 3. 2019's that scenario hydrolase is quite high, and 2027 it is expected also. Food and beverage segments held a dominant position in 2019 by exploiting the industrial enzymes, and would be maintain this lead in 2027 also. Here I have taken into one snap of the industrial growth of in India on the enzymes production. Novozymes India, they are established 1983. They are contributing 44% Indian markets. They're basically producing that hydrolase enzymes, protease, xylanase, amylase, and 30% contributed by the advanced enzyme technology. Rosary Biotech is recently, they are doing work for productions of the enzymes. So these are the scenario for Indian perspective. Now, already I told that if you think about productions of the enzymes, you need to concentrate on the systems which can able to produce huge amount of enzymes. Here we found that microorganisms contribute 88% for productions of enzymes. Animals and plants are less. Question is, why microorganisms? Definitely that question may arise, and microorganisms growth rate is quite higher. Specific growth rate is high, specific growth rate is very high, doubling time is very less. So in bioprocess, the microorganisms are used, have a short operation time, and the media component what you are using for productions of enzymes are very cheap, not so much expensive one. And you can change induce the microorganisms by manipulating the media. Most of the cases, people are doing several optimization technique to manipulate the media components. And also you can do the genetic manipulations of the microorganism too. Here is the disadvantage for plant and animal sources, where some cases you will get the potential harmful materials. These are the phenolic compounds that inhibits the productions of the enzymes. Although you can produce the industrial enzymes from the bacteria as well as fungus, bacteria, bacillus species exploit a lot for productions of industrial enzymes. However, aspergillus in the top, as it has several advantages. Not only it can produce the huge amount and the bacteria productivity is quite higher, but it can survive in the wide environments and can consume the various type of monosaccharide, polysaccharides, proteins, very complex compound. That's why it is a beauty for us to induce these compounds, to force the aspergillus to produce various type of industrial enzymes, that is protease, cellulose, xylanase, so that you can make it inducible, their enzymes are inducible in nature production. That you can do. Now, if you see that culture of the aspergillus basically is filamentous fungi. And if you take the spore and inoculate in the potato dextrose agar, you will see within 3 days the live aspergillus, that is white color, that's okay, it can be seen. That means it's a solid state fermentation in the media, in the solid phase is there. Spore took this food and germinate and produce the mycelia. But once you keep it for long times, you will see that again that mycelia is converted to spore formations, and that you can see under microscope also. At suitable environments, that spore will transfer to the germination tube, and that germination tube can be seen under microscope also. And after some times, if you supply proper foods and environmental temperature, pH, Then hyphae will be formed. And if you long time, if you keep, then that mycelia or long hyphae, complex will form. But these things are not suitable for productions of enzymes. These morphology cannot able to produce huge amount of enzymes. Now, if you think about the biological process or bioprocess development of productions of enzymes, we broadly classified that bioprocess in the two areas. First is the upstream part, and second one is the downstream. Upstream means up to biochemical reactions will consider from beginning, from raw materials collection, then pretreatments, then media formulations, then sterilizations, inoculations, and reactions, where your biomass and substrate utilization as well as product formations will take place. Whatever process you used, whatever process you adapt, up to the reactions, you can say this is a upstream work you have done. Downstream process is totally different. Here, no chemical reactions. Here, only the physical separation of the molecule of interest will be done. Once reaction is completed, downstream process is started. Here, normally the unit operations are employed. Filtration, centrifugation, precipitation, dialysis, drying, chromatography. These are, it's not that for all enzymes you have to use the same protocols. It's not possible because enzymes, molecular weight, the cueness of the amino acids, type of amino acid, that changes the uses of operation procedures. Here we are giving some informations what we did in our research lab for productions of enzymes. One beautiful enzyme, that is xylanase, from Aspergillus niger, and here three areas we have covered. If you keep the spore inside the refrigeration, under refrigeration, how long days should I keep? And when I'll go for the seed cultivation, should we go directly or I will keep long time inside the refrigerator so that manual cost can be minimized? So if you keep long time in the refrigerating conditions, then maybe your seed cultivation times will be longer, and then your recurring cost will be required more. And last one is the process optimization for productions of enzymes. Now we did several experiments where we preserved the spores at different conditions, not different conditions, different times in under refrigerating conditions. What we observe, if you keep for 28 days, the spore size is automatically increased, because you know that spore having some foods that the normal work maintenance purpose. But if you increase more times, then its size will be reduced, and then it's detrimental for the spores. You cannot go beyond that. So here 28 days is recommended once you preserve, and after go directly for the seed cultivations. So in seed cultivations, basically when we do, why you are going for the seed cultivations? Because you know in the reduce the lag phase, when any microorganisms grow, when they face a new environments, they took some time to understand the environment, and therefore is a question of lag phases. And that lag phases, to reduce the lag phase, we design the seed cultivation process. Here we worked on several pHs and several temperature, and we find out what is the length of the germination tube, what we observe. And that is given here, and we found at pH 7, if you see that germination tube's length is quite higher after 18-hour seed cultivations. And that is evident also at 28 degrees centigrade, 7 pH. But it's very difficult every time you'll go for doing the seed cultivation, go for microscopy, understand the length, average length you have to take. It's very difficult. Therefore, if you see that various time interval, whatever we took the samples from the seed cultivations and go for microscopy, the germination tube's length is elongated and is found maximum when the temperature is 28 degrees centigrade, pH is 7. If you go further, then that high P will be transferred to micelle. Is very difficult to control in the production media. Because I already mentioned, this morphology never give any enzymes higher amounts. But every time if you go for microscopy and see the germination tube length, it's very difficult. Therefore, a marker has been incorporated. The pH, if it is below more than 6.2, it should be within 6.2. If it is less than 6.2, that means high P conversion, the micelle conversion can take place. So therefore, 6.2 is considered as a marker for seed cultivations. Now, when you are talking about the batch cultivations, once seed cultivation is completed, and you are thinking for the batch cultivations, normally we people use a bioreactors. Bioreactors means nothing different from the reactors. It is a reactor where the reaction takes place. It's a vessels where the biochemical reactions takes place. But the difference is from the chemical reactor to biochemical reactors. Biochemical reactor, that acceptability maintained. Aseptic condition you have to maintain because some opportunistic bacteria may grow. Not only that, when we design a reactor on the basis of reaction engineering perspective, we consider the reactor has uniform distribution of temperature, pH, fluids, and mass. When we do the experiments in the small scale, then it may not affect a lot. But when you are working with the reactor in the big vessels, mass transfer play major roles, and it is critically hampered the overall productions of the enzymes. As you know that elementary rate law, that is dP by dt, that is product formation rate, is K into P. So that P or dx by dt is equal to K into X. X is the biomass. So that biomass should be uniformly distributed. When you are designing reactors, that assumption was taken, and ideally it is considered the mass should be uniformly distributed. But practically, mass is not uniformly distributed. Therefore, batch to batch variations of the products, to minimize that, you have to think about that a mixing device need to be incorporated. What is that? That is the impeller. Impeller is basically a agitator which helps to mix. But problem is that when you are using the impeller, that will create the vortex inside the systems. Vortex means the mixing is not having proper. Then to remove the vortex, you need to think about vortex remover. It's called the baffles. That's why if you see in the reactor, the size in the various sizes, if you see the reactor, the baffles have been given inside the reactors to remove the vortex. And what will be the size of the impeller? What will be the clearance of the impeller? And how many impeller need to be incorporated? That depends on what is the clearance of the impeller. If you see the mixing agitator, a impeller, when it works, its movements, then only you can see. As well as when a biochemical reactions needs oxygen, because most of the cases is aerobic process. So you have to give the oxygen from outside. That means you can give the air, you can sparse, and that demand of microorganisms demand on the oxygen. You know that oxygen is sparingly soluble in the waters. It's 8 ppm hardly. So bubbles should be retained longer time inside the reactors. That's why the length of the reactor is very important. If you increase the length, then only the bubble can stay longer time inside the reactors. If it is sparse, its area is very small, and its height is reactor's height is more, then it can be maximally utilized. So design of the reactor case to case will vary. Aspergillus niger, if you use the needs oxygen enormous amounts. So you have to think about design. When you are designing of reactor, you are thinking, then height of the reactor should be more. Now, if you go for mixing, what is happening inside? It may be the that radial mixing that's happening here. This is impeller. You just see when it moves clockwise, the impeller start to pump the waters and throw it, and just throw is a 90-degree angle with its axis. It's a radial impeller. Once it's pumping, that liquid it hit the wall and distributed to two directions. And both directions, it's create the mixing capability of the enzyme in the agitators. So that's the clearance, the height of the liquid, which can be mixed properly by the impellers. So that's is very important, but you cannot decide the impeller what without blindly you can take one impeller. No, this is a biochemical reactions where you are dealing with living systems. You are dealing with Aspergillus niger. You are dealing with the bacteria. So it should not damage the cells when it's moving inside the reactors. The fungi should not get any damage from the impellers. If get damaged, then there is a question of productivity. That's why people use the Ruston type turbines. You just see its three-dimensional view. When it moves, just see that when the fungus, neither it hit the walls, it basically kissed the walls. So that design of impeller is very, very much important when you are using any kinds of microorganisms mediated production systems. Impeller, not only the function is the mixing, but also it affects the KLA, volumetric oxygen transfer rate inside the systems. Obviously, Ruston type turbines needs more energy, electrical energy, to pump the same amount of liquids. There are lots of impellers can be used in chemical reactions, but Ruston type turbines, 6-blade Ruston type turbine is used for the biochemical reactions. This is one more problems. When you start the biochemical reactions, you will see that your impeller, initially it's run, but after sometimes stop to run. Because you start the sparging of the sparging by the air. So what will happen? That air creates the bubbles and that blanket the impellers. So impeller cannot move. It's the flooding cases of the impeller. The impeller is not supposed to move because the bubbles are blanketed on the impellers. So you are expecting in the small scale that problem can be minimized. But in the large scale, when you are going for productions of any compounds, not only enzymes, your bioactive compounds, then you will face these problems, and that can be solved. If you increase the speed of the impellers, then impellers start to move. Is called the loading situation. Then after that, if you increase the impeller speed, then it will create the complete dispersion. Here you can see that figure C, where the complete dispersions of the bubbles can take place, and you already mentioned oxygen is sparingly soluble in water. So oxygen should be distributed uniformly throughout the reactors. Microorganisms which is present in the systems, every corner they can utilize the oxygens. Already, if you see the stat tank reactors, where oxygen amount, how much oxygen is there dynamically, you can calculate, you can measure by the DO probes. DO probes is the probe which is present in the reactors, which can dynamic measurement of oxygen can be done. Another important probes that is used in the reactors is basically pH probes, and that pH probe will help to understand the pH of the media, what is present. I already mentioned catalytic reactions, that may be the microbial or that might be the enzymatics. The pH and temperature of the environment need to be uniform. Another very important part is the temperature probes that also used to maintain the temperature of the systems. If somehow any reactions is going to happen, is exothermic, that heat is generated. To remove the heats, that chilling systems are also incorporated in the during design of the batch reactors. We can use the stat tank reactor as a batch reactor because there is no input, no output during the reactions. So once the reaction has been started, then you cannot add any substrate during the process. As a whole, you will understand the concentrations of substrate is coming down, and that's fungi will get a stress that substrate is going to contribute. Here, I have shown you, already I have told that oxygen is sparingly soluble in the waters. Now, retention time of the oxygen bubbles will tell you the necessity of microorganism for their respiration. So if the bubble size is small, they will remain longer times, and they will take long time to leave the reactor's passage. And there are several 8 steps are involved. It is a different phases reactions. Your bubbles in the gas stages, in the air bubbles, and the bubbles, the oxygen need to be passed from the bubbles to liquid, and there is a resistance. Because when two phases meet, a resistance creates called the boundary layers. That gas liquid interface that can be minimized by increasing the agitation speed, but not always. If you go for, then there is a chance of cell damage. Viscosity of the media when go for the fungal fermentations, definitely the viscosity will be enhanced 100 times, 10 to 100 times. So once the viscosity is enhanced, then the oxygen solubility will decrease. So that's the problems that need to be maintained by the that agitation and aeration regime. That is called the volumetric oxygen transfer rates. Here the liquid will move, oxygen will move from the gas bubbles to the liquids, and that resistance can be minimized by agitations. Then is a liquid phase, either in the cell is in the clump conditions, or in the microbial cells or in the single cells. That depends on that liquid to solid oxygen transfer will take place. And it is an 8-step, but most of the cases, the resistance is here. And that resistance can be minimized by increasing the agitation speeds only. In our case, we optimize the whole batch process, and we found that carbon source xylenes need 3 gram per liters, and potassium nitrate need 10 gram per liters. Sodium chlorate 5 gram per liters, magnesium sulfates, inoculum percentage 5%, temperature we need to maintain 28 degrees centigrade, pH 7s, agitation 150 rpm, and the xylenes production was found 1625.74 unit. Although nowadays people are using several statistical tools, because change in one variable at a times method is not sufficient to get the real optima or global optima, which basically needed for engineering point of view. Now people use that several methodology like response surface methodology, design of experiments, the box pattern designs, Taguchi methodology, artificial neural network. So whatever they are using, ultimate goal is to find out the optimum, real optimum conditions which can be utilized in the bioprocess to get the maximum output. Once we did the batch process, then we have a curiosity. Can we increase the content amount of the enzymes? Yes or no? Then to understand the behavior of the process, when you are going to design a process, first of all, you should know that reaction engineering perspective, what is the kinetical behavior of the systems. Although we did biomass growth kinetics, substrate utilization kinetics, and product formation kinetics, but here we will only mention that product formation kinetics because it is related with the product, that is xylenase. 2000, Ludeking and Piret, both scientists, gave a model that when any biological product is found, that follow the equations, and that equation is not new, it's very old methods. Here that dx by dt is equal to dp by dt is equal to alpha into dx by dt plus beta into x. So here our intention, significance, that product is growth associated or product is non-growth associated. Then you make a plot with specific growth rate versus specific production rate. From that, you can understand the value of alpha and beta. If you get the alpha value is there, then it is a growth associated events. That means your enzyme is producing during growth of bacteria or growth of microorganism, growth of aspergillus. If you get the beta value is 0, alpha value is 0, that means it's happened in the stationary phase. It is a secondary metabolites. If the alpha and beta is not equal to 0, then you can say it is a mixed product. That means either it can produce during the growth or it can produce in the stationary phase. In our case, when we plot the data, whatever we got from the experiments, and we surprised that it is happens in the stationary pages. Then we understand the utility of the fed batch process. As you know that when the antibiotic productions came to the market, that is penicillium notatum. When it is industrially produced, production takes place. In that time, people tried a lot in the batch process. They failed because of the batch process. I already mentioned that batch process where you have given the food initially huge amount, works under unsteady state, the substrate will going to consumed, and fungi, they use the foods and produce the acid, and that inhibit the growth. And here also same thing. Same observation we found, and that's why we went for the fed batch process where steady state can be maintained. What you got in the stationary page where the growth rate is 0. That can be maintained in the fed batch process. In the fed batch process, basically fed batch process, if you think about fed batch process, it is works under quasi-steady state. What is the meaning of that? Meaning is concentrations of biomass, concentrations of substrate, concentrations of product will remain constants, which is never possible in the batch process. That means we are giving food at certain concentration to the fungi and creating the shock that food is not enough available for your enjoyment or for your growth. Only you think that this food is providing to you for your normal activity, and it is a shock to you to produce the enzymes. That secondary metabolites all produced in the stationary page, and that's why we plan to give the shock to microorganisms by 3 ways. First one, will give the food limited amount initially, and slowly will give the food so that fungi can face stress constantly. Second one, will give the stress mechanically. Not only food, I'll give the mechanical stress to you. And third one is the pH. Will change, will alter, will give the pressure, stress by changing the pH of the media. We start our observations, and what we found, we just see when that xylence, what we did initially, if you remember, that initial amount was 3 grams per liter in the batch. But when we start the fed batch process, then we give 1.5 grams per liter. So 50% of the xylence, that is carbon source, which is required for their growth, that amount we have given in the initial stages. So after that, what we observe? What observation you see, if you see carefully, that xylene is going to, carbon source is going to approaches to 0 after 72 hours. That means we are giving up to 72 hours, we are giving to you for your enjoyment, for your growth. But after 72 hours, you will get the huge shock. That means you go for the production, it's a threat to you. And if you see that agitations, when you start the agitations, it is a 120 RPM, and then it is continued to 12 hours. That is, then again we increase the agitation speed 150, and then it's continued to 48 hours, and then again we increase the speed because the viscosity of the media is changing. Once fungal start to grow and form the mycelia, the medium become more thicker, and that is more dense, so that the viscosity of the media becomes changes. And if you Do not increase the agitation speed. The all fungi will die because oxygen limitations will come into picture. So you have to increase the speed. For our intention is to not die, to kill the fungi. You have to give the certain amount of foods, and that we have given at 72 hours. You see that when food is given, then again concentration is increased, and up to 120 hour again we are given the feed, and then constant concentrations of the xylins we are maintaining here. That we are not initially like batch process giving the huge food. No, we are constantly giving so that concentrations of the carbon source is remain constant. That's we need to maintain. And agitations, I already mentioned one thing when you start the discussion, that to handle the fungi is not a very small things. It creates a lots of problem in the during fermentations. Neither germination tube, neither high P mycelia, they never produce the huge amount of enzymes. You have to transfer this mycelia to the beads or pellet forms. Only pellet can produce the enzymes or secondary metabolite. So what we did, up to 72 hours, we give the enough scope for fungi for their growth. But after that, we increase the speed, that is 210. So impeller is moving at 210 RPMs. So that RPMs, fungi's mycelia is chopped into small pieces, and that piece create the pellets. And it happens after 72 hours. And it will continue. This process was continued up to 144 hours. You just see 210, then 240 at 120 RPMs. Complete conversions of the mycelia to the pellet formation takes place. Once complete pellet formation takes place, then there is no need of extra energy. You just minimize the RPM of the agitations, and your pH is changing, looks like. If you see carefully, the role of the pH, to understand the role of pH, we did the experiment at various initial pH. What we observe, microscopically what we observe, that pH, when we run the fermentations at pH 7.5 and 7 and then 6.5, we found that more complex networks of fungi we got at pH 6.5. And this fungi, it is very difficult to chop into small pieces. Not only that, it's create a thick environments that is difficult to control during the process. So we take the pellets at 72 hours, and we found that the pellet size at the beginning 6.5 is very high, but when we came to 144 hours, the pellet size is quite similar for 7.5 and pH 7. But the pellet size is equally important, as well as the number of pellets. That will tell you how much amount of enzyme to be produced. So here our observation is that when we start the work at the initial pH 7, then the number of pellets as well as the size of the pellets both are higher. The number of pellets are higher in case when you start the experiment at pH 7. That's the observation we did, and we published 2 to 3 papers also from there. And that is the selections when I will decide at which pH, which temperature will do the works, and that help to understand, to design a big commercial available process. When we compare the batch and fed batch process, what we found that both cases we got the 7 days experiments, and here 2000 here we got the 2524 units. Now, definitely once your reaction process is over, you can go for the dialysis, ammonium sulfate precipitations methods usually done is external enzymes. Go for the simple centrifugation process. Your fungi will be separated and your enzyme will be in the supernatant. Since it is a soluble in waters, so you just do the centrifuge, remove the solid precipitate and take the supernatant, go for the ammonium sulfate precipitation. So you know this is called the salting of defect. You add the ammonium sulfate, protein will precipitate. It does not mean that if you add, it will precipitate. Sometimes it may not precipitate. So it depends on the balance of energy. What about the games here? That is only on basis of energy. How much hydrogen bonds are hampered due to addition of the salt, that will decide who will precipitate, who will not. What we got at 60 to 80% ammonium sulfate precipitations that have an in 81% in these fractions. But before going to understand the activity, you have to go for the dialysis, because the ammonium sulfate precipitation, when it is going to happen, then proteins is also precipitate along with the ammonium sulfate. If you do not remove the ammonium sulfate, protein will not show its activity. When I start the discussion, I told that its structure will say only its function. If somehow you disturb its structure, it will never show the functions. Once ammonium sulfate is there with proteins, its three-dimensional structure is critically hampered and it will never show any activity. You have to go for the dialysis. Most of the cases we use the membrane, 12 kT membranes, that is 12 kilodaltons. 1 dalton is equal to 1 into 1.66 into 10 to the minus 24 grams, means 1 by Avogadro numbers. That amount of biomass, that molecular mass can only be retained, and more than that can only be retained, and less than that will be removed from the dialysis bag. You know that ammonium sulfate daltons is very less. So ammonium sulfate, once it is coming out, your protein of interest will get the life, because it's again comes to its three-dimensional structure and it starts to work. Now, another important part our students did, when sometimes you will may not get the purified forms of the enzymes after ammonium sulfate precipitations. So let's do for the chromatography. Here you will go for the that is size exclusion chromatography. You will see that here this fraction is only the impurities, but these are not the proteases, the enzymes. Here is the case, this fraction having the protein contents. Here is a proteins as well as protease. So there is a possibility to get the purified form of this protein here. So take this illusion you take and go for the ion excess chromatography. And here also, if there is a impurities, that can also be separated. Here this fraction you only get the proteins, that is impurities, but your protein of interest is here, and that can be easily separated using the ion excess column. Now this is that size exclusion chromatography, SDS page sodium dodal sulfate polyacrylamide gel electrophoresis. So electrophoresis, here you can get the estimated molecular weight of your proteins. So you'll get that 60 to 80% fraction. This protein is around 63 kilodaltons. That proteins. So this is your markers. So you will get approximate molecular weight of your proteins. Now, when a protein or when enzymes you are going to purify, you should keep in your mind that enzymes should be activated at robust temperature. Either its importance towards application will be very limited. Its temperature, high temperature should work, and a broad range of pH is also important. Here the temperature, that is 50 degree, what we got, our enzymes active at 50 degrees centigrade, And the broad pH range from 2 to 5, then we can use for any catalytic reactions. You can see that if you increase the pH after 6 to 12, its catalytic efficiency is drastically reduced. Now, we did one patents, not only the enzymes activity, but also its stability. It should stable at the environments. But we found that pH 2 to 5 in that region, that enzyme can stand without losing its activity for 1 hour, and that we did and go for patents, and it's luckily it's patent in 2021. Then we thought, why you cannot apply these enzymes for the fruit juice clarifications, because the most of the fruit juice having the pH range within 4. So why our enzyme cannot be used for the clarifications of that? What we got, interestingly, what we used the 2000 unit per ml of enzymes, and we got the lemon clarifications is quite higher, 78%, then pineapple, then Mosumbi. What our observation that lemon contains more amount of xylene. So this is a snap that is controls and the blanks. That part we have shown you. These are few publications that we did in the coming last 5 years. That Uma Shankar Prasad, that basically he did the work on the xylenase. Now is the faculty of the Benware, and this is another student's work on the bioremediations. Rambalak Mahato also did the work on the pectinase, and Payal Chaudhary did the work on the microbial fluorescence. Present students, they are working in various fields. In the field of the biotechnology, bioinformatics, and some people are working on the food technology. Thank you for your kind patience. So any query, please.
Yeah.
Hello.
I request Lakshmi Satya for the question. Yeah.
Yes, ma'am.
Anyone?
1 minute. Am I audible?
Yes, ma'am.
Yes. One of our colleagues for required pellet consistency, or it changes from case to case.
Pardon, madam. Please repeat.
Yes, sir. What is the criteria to determine RPM for required pellet consistency?
No, it is very difficult. That RPM you cannot fix, because already I told madam that your intention is to produce the enzymes, but it should not be in the micellar stage. So if you convert the micellar to pellet, then definitely your agitation speed need to be enhanced. If you do not enhance, your micellar will remain as it micellar forms. So our intention is not that to keep the fungus in the micellar form, because micellar never produce any enzymes. It produces the less amount of enzymes. You need to give the shock, and that's why you need to increase the RPM for the fungal fermentation. But in case of the bacterial fermentation, there is a consistency you can maintain. But bacterial fermentation speed not so much important to control the create the stress. Okay.
Yes. Sir, there is another question from one of our students, sir. So for enzyme extraction, when you are isolating microorganisms, is it better to go with fungal or bacteria?
Madam, I suggest if you able to handle the fungi, it is best, because titer will be very high in fungi. Aspergillus, they will give in 7 days, 2000 units or 2500 units, that you never get from the bacteria. If luckily you can do for the recombination or you can do some mutations, then that is a different. But handle the fungi is very difficult. If you could able to do, then that's fine, that you go for fungi.
Let me check, sir. Sir, there is another question in chat box. What is the reason behind to select only those 3 fruits? You told us, sir, lemon.
Actually, thing is that already we told that our enzyme is stable and active at the pH range 2 to 5. So in northeast regions, we choose the fruits what are available here, which can pH of this juice are within pH range from 2 to 5. That only we have taken. It is not compulsory that only these 3, it will work on 3 fruit juice. No, only that for testing purpose, we took 3 juice which is available readily at the northeast region.
Thank you, sir. Thank
Okay.
you. Valuable.
Thank you. Thank you very much for giving me opportunity once again to all people's managements, the faculty members of the VIPER, especially madams and Dr. Wanteri sir. Thank you. Thank you very much.
Yes, sir. Wait. Sir, I request Lakshmi sir to propose vote of thanks.
Okay.
A very good afternoon to the respected dignitaries, principal, faculty, delegates, and my dear students. Myself be Lakshmi Satya, Associate Professor from Pharmaceutics Department, VIPER. It gives me immense pleasure to honor vote of thanks for the Day 4 session of FDP on recent advances in science and technology innovations. On behalf of VIPER, I would like to thank Dr. Vishwanath Bunyath sir for his excellent lecture on enzyme isolation and correction. I thank to management for trusting us and always giving us pleasure to conduct such events. I thank our principal sir, Dr. A. Ramesh sir, chairman of this event, who always behind us and motivate us to do such programs. My special thanks to Dr. O.N. Tiwari sir, principal scientist, ICAR, IARA, New Delhi, and co-chairman of the FDP. I extend my thanks to our HOD, Dr. K. Vanitha ma'am, and also to the coordinators, organizers, Team VIPER, and whoever involved in this program. Last but not the least, I'm thankful to all network supporting team who plays a major role for the success of this event. I thank all the faculty, delegates, and my students and other students from various colleges of India for participating in the event and making the event a very grand success. At the end, I would like to request all delegates to fill the feedback form and give us the suggestions regarding the session. I take leave with a hope that we will meet tomorrow with a lecture by Dr. Rangareddy Burri sir with same zeal and enthusiasm. Thank you one and all. Thank you, sir.
Thank you. So,
Thank you, sir.
Madam, can I leave? If you
Yeah, please, sir. Thank you, sir.
Okay. Thank you. Thank you very much.
Thank you, sir.